Okay, so again, we are going to do a workshop on the Hardy-Weinberg equilibrium. The Hardy-Weinberg equilibrium is an equation that is used to analyze genotypic frequencies and allelic frequencies in populations in nature. And so, in order to be able to do these types of problems, there are some vocabulary terms that you need to understand. And so we're going to go over those first. Um, the vocabulary is the homozygous dominant, homozygous recessive, and heterozygous terms. And when we talk about finding genotypic frequency or allelic frequencies, we need to understand what it means to be homozygous dominant or homozygous recessive or heterozygous. And so um, genotypes are the combinations of the alleles that are inherited by an organism. So if an organism is homozygous dominant, that means that they have inherited two dominant alleles from their parents. So one dominant allele from one parent and the other dominant allele is inherited from the other parent. And so their genotype would look like this. It's going to be two dominant alleles represented by a capital letter, right? So two capital T's. We're going to use T's just because they're easy to tell the difference between capital and lowercase. So for the homozygous recessive, this means that they inherited two recessive alleles, one recessive allele from one parent and another recessive allele from the other parent. And so their genotype would be little t, or two lowercase t's. Now, if an organism is heterozygous, that means that they've inherited a dominant allele from one parent and a recessive allele from another parent. Okay? And so we can write these alleles over here. The dominant allele is represented by a capital T, and the recessive is represented by a lowercase t. Now, one of the important things to be able to do first is to be able to relate the variables in the equation to the genotypes and the alleles. And so we know that the dominant allele is T, and that corresponds to the P in the equation. So we're just going to write a little P here because we know that that's what represents the dominant allele. And then for the recessive allele, that is represented by Q in the equation. And so I'm going to write a little Q here. Okay. And so we know that the recessive allele is represented by Q. Now, down here we have P squared and Q squared. So P squared is going to be representing the homozygous dominant genotype. So this would be a genotype. So this would be big T, right? And then the homozygous recessive is going to be represented by Q squared, right? And then the heterozygous, so this is TT. So the heterozygous then is represented by this 2PQ here in the middle. And so in order to figure out the heterozygous genotypic frequency, we have to take 2 times P times Q. So you have to know both P and Q in order to figure out the frequency of the heterozygous individuals. And so I'm going to just write 2PQ here. Okay. So hopefully that helps you to kind of see how our genotypes and alleles can relate to the equations that we're using to solve these problems. So let's go ahead and look at this first problem. Make it a little bit smaller. It says allele T for the ability to taste a particular chemical is dominant over allele little t for the inability to taste the chemical. So little t, little t is for non-tasters, right? So let's write that out. This is what they're giving us in the problem. Little t equals the non-tasting allele. And then the capital T is the tasting allele. Right, so there's our two alleles. 400 university students were surveyed and 64 were found to be non-tasters. So 64 out of 400 were non-tasters. In order for a person to be a non-taster, their genotype must be homozygous recessive, right? So TT. And we know that the frequency of this genotype in the population can be calculated by taking 64 divided by 400. So in order to find TT, we need to take 64 divided by 400. And so that equals 0.16. So now let's start putting this in our equation. And one of the hints I want to give you about Hardy-Weinberg is that the easiest equation to use is the P plus Q equals 1. And so that's the equation that we're going to use first. But first we need to get this out of this form, right? And into just the T form. So right now we know that the homozygous recessive genotype has a frequency of 0.16. So if we go back to our other slide, we know that Q squared is equal to that homozygous genotype. And so we can also put that equal to 0.16, right? So if Q squared equals 0.16, and we want to use this equation, P plus Q equals 1, we need to find Q. And so in order to find Q, we're going to take the square root of Q squared and the square root of 0.16, and we get Q equals 0.4. So now, because we're looking for heterozygous students, we know the allele frequency for T, right? But we need this. We need 2PQ, right? Because that's the heterozygous genotype. And that's what they're asking us to find. So what we do is we find P by rearranging this equation. So 1 minus 0.4 is going to equal P. So 1 minus 0.4, we know P is going to equal 0.6. So now all we have to do to get the heterozygous genotype is plug 0.4 in for Q and 0.6 in for P. So let's do that. So 2 times 0.4 times 0.6 equals, and remember, 0.4 corresponds to Q, and 0.6 corresponds to the P. So what do we get? We get 0.48. And so that's our answer, right? 0.48 would be the, and now we have to calculate the percentage. So we just have to turn 0.48 into a percentage. And so we know that the percentage of heterozygous individuals is 48%. So let's do one more, right? Whoops. Or, good. Oops, I don't want to do that. Okay. So this is about corn. It says in corn, purple kernels are dominant to yellow. A random sample of 100 kernels is taken from a population in Hardy-Weinberg equilibrium. It's found that 9 kernels are yellow and 91 kernels are purple. And so for this one, we're going to use a capital K for the purple, since that's dominant. And we're going to use a lowercase k for yellow. All right. And what is it asking us to find? It's asking us to find the frequency of the yellow allele. So we're looking for an allele frequency this time, not a genotypic frequency. So what are they giving us? We have a sample of 100 kernels, and they found that 9 are yellow. So we know that 9 must be, in order for them to be yellow, they have to have 2 lowercase alleles, or they have to be homozygous recessive. Now we're going to do exactly the same thing as we did in the first problem. So we know that KK, or lowercase k, is equal to 9 out of 100, which is equal to 0.09, right? And that is going to also equal, since we know that in our equation, P plus Q equals 1, and P squared is equal to our, oops, we don't want TT, do we? We want KK. So that's capital K. Capital K, Q squared equals lowercase. And so Q squared is going to be equal to 0.09. So in order to get our allele frequency for the recessive allele, all we have to do is, so we have Q squared equals 0.09, right? We have to take the square root of both to get, whoops, we have to take the square root of both of these in order to get Q, which is what we're looking for. I don't know why these things keep popping up. Sorry about that. So let's do that. Let's do square root, and so now we have Q equals 0.3, which is the square root of 9. And so our frequency of the recessive allele is going to be 0.3. We can also write that as 30% if they ask you for the percentage. And that's very crooked. Sorry about that. All right, well, I hope that helps. If you have questions, you can jump into the question and answer part of the workshop coming up. Right? Okay.